So dear students, welcome in this e-content of the second year of B.sc. So today we are going to discuss a very vital topic in which we will undergo some important information about glycolysis. So it is coming under the respiration. So generally, we can define respiration as it is a cellular metabolic process. So it is happening inside the cell. It is a biochemical process. So it is cellular metabolic process wherein the living cells of an organism, any type of cell is there. So respiration is happening in every type of cell. So that cell obtains the energy in the form of ATP by the oxidation of complex organic substances like glucose. In that process, oxygen may be involved or may not be involved. So this brings about the aerobic or anaerobic respiration. Whenever there is involvement of oxygen is there, so that is called aerobic respiration, while where no involvement of oxygen, that type of respiration is called anaerobic respiration. That is also called fermentation. So, and in this process, oxygen aerobic respiration, in aerobic respiration oxygen is taken in and carbon dioxide is given out. So this process of aerobic or anaerobic respiration, it principally firstly involves the step that is called glycolysis. That pathway of glycolysis is also referred to as Embden-Merov-Parnas pathway. So three different scientists are there. They have given this pathway of glycolysis. So glycolysis is cellular metabolic process in which one molecule of glucose is broken down, enzymatically broken down. तो एक मॉलिक्यूल जो ग्लूकोज का है उसका ब्रेक डाउन होता है enzymatically step by step. So this glycolysis is a 10-step reaction. There are 10 different reactions involved in this. So ultimately one molecule of glucose is broken down to form two molecules of pyruvate, and also two molecules of ATP and two molecules of NADH are also produced. So let us start with this one molecule of glucose. Here it is given that one molecule of glucose in this first reaction of glycolysis, it is converted into glucose 6-phosphate. So here is glucose, it is being converted into glucose 6-phosphate. So there is addition of phosphate group. Enzyme working in this reaction is hexokinase. हेक्सोकाइनेस एंजाइम काम करता है रिएक्शन में. One ATP will be utilized and that will be converted into ADP. So hexokinase enzyme transfer the phosphate from ATP to the glucose. Ultimately at the 6 number carbon of glucose, one phosphate group will be attached. This hexokinase enzyme requires magnesium or manganese ions for their enzyme action. This first step is called phosphorylation, where the addition of phosphate into the compound is taking place. Next point, next reaction is the production of fructose 6-phosphate from glucose 6-phosphate. So this is generally a reaction of isomerization. So glucose and fructose are isomers of each other. Enzyme working here is the phosphoglucoisomerase. Phosphoglucoisomerase is working, and this is a very simple reaction. We can refer to it as isomerization. In next step, fructose 6-phosphate is converted into fructose 1,6-bisphosphate. So simply what is happening here, one additional phosphate group is being attached to the first कार्बन ऑफ द फ्रुक्टोज. फ्रुक्टोज 6 फॉस्फेट के ऊपर एक नंबर के कार्बन को और एक एडिशन हो रहा है फॉस्फेट का. सो प्रोडक्ट अल्टीमेटली फॉर्मिंग इज fructose 1,6 bisphosphate. There is presence of 2 phosphate. This reaction, enzyme working in this reaction is phosphofructokinase, which a very vital enzyme. ग्लाइकोलिसिस का रिएक्शन जो है इस एंजाइम से भी रेगुलेट किया जाता है. सो हियर इज इन्वॉल्वमेंट ऑफ 1 ATP molecule which donates the phosphate, and phosphofructokinase adds this to the fructose 6 phosphate by forming the fructose 1,6 bisphosphate. Here, for activity of enzyme phosphofructokinase, magnesium ions are very important. This reaction is referred to as phosphorylation. Phosphorylation are those reactions where addition of phosphate is taking place. Next step is the conversion of fructose 1,6 bisphosphate to the some spelling mistake is here. So consider it as fructose 1,6 bisphosphate. So fructose 1,6 bisphosphate is being converted into fructose 1,6 bisphosphate is being splitted. This is reaction of cleavage. So one molecule of fructose 1,6 bisphosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. As ultimately fructose 1,6 bisphosphate is having 6 carbons in one molecule. So there are 2 molecules formed, one molecule of glyceraldehyde 3-phosphate. This is also referred to as 3-phosphoglyceraldehyde alternatively, and one molecule of dihydroxyacetone phosphate is formed in this reaction. So every molecule is of 3 carbons. So total carbon number will be maintained at 6. Enzyme working in this reaction is aldolase, which requires zinc ions for its activity. This reaction is called cleavage. Again, this is reversible reaction where enzyme is working triose phosphate isomerase. So simply it is a reaction of isomerization where glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are being interconverted to each other. So this is also a reaction of isomerization. So 5 reactions we have till covered. These 5 steps are referred to as payoff phase. These are payoff phase where ATPs are being invested, ATPs are being utilized. ATP का खर्चा होता है, एनर्जी का खर्चा होता है. Next, ultimately product formed glyceraldehyde 3-phosphate is utilized in further reaction 6 to 10. So let us discuss about that. So glyceraldehyde 3-phosphate, which was the last product of 5th reaction, is being converted into 1,3-bisphosphoglycerate. This is reaction of dehydrogenation where hydrogen is being transferred from the glyceraldehyde 3-phosphate. So enzyme working in this reaction is glyceraldehyde 3-phosphate dehydrogenase. So name of enzyme is coming from the substrate itself. So there is formation of 1,3-bisphosphoglycerate, that is also called 1,3-bisphosphoglyceric acid. Here is hydrogen group is removed from this glyceraldehyde 3-phosphate. So ultimately compound formed is 1,3-bisphosphoglycerate. We should remember that here 2 molecules of NAD plus or oxidized NAD form is converted into 2 molecules of NADH. So here energy is being formed. Next, in 7th reaction, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. So by casual observation, it is clear that here is removal of one phosphate. So one phosphate is being removed. Enzyme working in this reaction is phosphoglycerate kinase. This is a crucial step in the glycolysis, and this is called substrate level phosphorylation. Why this is called substrate level phosphorylation? Because there is production of two molecules of ATP. So ADP is converted into ATP, adenosine diphosphate is converted into two molecules of adenosine triphosphate. So this is a chemical energy that is being formed, generated. Next reaction in the 8th step is just movement of phosphate group from 3 number carbon to the 2 number carbon. So there is just shift of phosphate molecule. So compound formed is the 2-phosphoglycerate. 3 नंबर कार्बन से निकल के फॉस्फेट जाता है 2 नंबर कार्बन के पास. So enzyme working in this reaction is phosphoglycerate mutates. Enzyme is typical, but reaction is of isomerization because no molecular change is taking place. Next to that, the last product formed, that is the 2-phosphoglycerate, is getting converted into phosphoenol pyruvate, that is also called phosphoenol pyruvic acid. So ATE in all over this reaction can be replaced by IC acid. So we can refer it as phosphoenol pyruvate as also phosphoenol pyruvic acid. So one important step where dehydration is taking place. This reaction is called dehydration because here is removal of two water molecules taking place. Enzyme working in this reaction is enolase, which requires magnesium or divalent manganese ions for its activity. Next to that, phosphoenol pyruvate is finally converts into pyruvate, and there is a key enzyme that is called pyruvate kinase that also requires magnesium ions or potassium ions for its activity. Here in this reaction, two ATPs are formed. So this is also called substrate level phosphorylation. So we should remember that in this total process, net energy production is 2 ATP and 2